Welcome to the Longevity Papers podcast, where we delve into the latest research shaping our understanding of aging and health span. This week, we're looking at a fresh stack of papers, really focusing on what's new and noteworthy for biotech researchers like you. We're trying to sift through the noise, find those actionable insights.
Yeah, and to be frank, it's been a pretty typical week in longevity research. Maybe 95% of what we see, it's incremental stuff, not really those groundbreaking paradigm-shifting discoveries we're all hoping for. Most papers are exploring known mechanisms or offering modest refinements, adding bricks to the wall, basically.
So no seismic shifts this week then, more like crucial refinements, adding detail.
Exactly. But that said, we have picked out a few papers that are genuinely worth talking about. Though, as always, we need to approach them with appropriate skepticism about, their truly transformative potential right off the bat. Our mission is always to pull out the most important nuggets, highlight surprising facts, give you that shortcut to being well-informed. This week, we're seeing themes around how different systems connect, and inflammation seems to be a recurring thread.
Okay, let's jump into the material then and unpack what really stands out. We're starting with a paper that touches on a classic longevity intervention, caloric restriction. The title is Exoproteome of Calorie-Restricted Humans Identifies Complement Deactivation as an Immunometabolic Checkpoint Reducing Inflammaging.
Right, this one's interesting. It's on bioRxiv, that's pronounced bioRxiv, posted August 6th, 2025. From Mishra, Kim, Yom, and colleagues at Yale School of Medicine. The core idea is that caloric restriction, CR, seems to work at least partly through an immunometabolic mechanism, specifically modulating the complement system. And crucially, they did longitudinal plasma proteomics on humans undergoing a pretty significant 14% CR for 2 whole years. That's human data, which, immediately makes it more relevant than just cell culture studies.
Okay, let's dive into the specifics there. What exactly did they find happening in those people on CR?
Well, what they found was a significant lowering of a protein called C3a. Now, C3a is a key part of the complement pathway. Think of it like the immune system's emergency alarm. When it's overactive, especially C3a, it fuels inflammaging, that chronic low-grade inflammation that drives aging. And they saw this reduction in C3a correlated directly with lower inflammaging markers in the CR group. It suggests C3 deactivation is maybe a metabolically controlled checkpoint.
So a specific molecule, C3a, affected by CR, and it's tied directly to this big problem of inflammaging. That feels like a strong connection.
It is. What's fascinating is identifying C3a as this specific target. It looks like it could be both a biomarker for healthy aging and a potential therapeutic target. The human data, like I said, 14% CR over 2 years, that's solid. But they didn't just stop there. They then went into mice. They used specific small molecule inhibitors against C3, and that inhibition reduced inflammaging, improved metabolic health, and importantly, boosted health span in the mice. That really points towards a causal link, between C3a and these aging outcomes.
Okay, that sounds pretty significant. But stepping back, how truly novel is this finding? I mean, CR research goes way back to the 1930s. Clive McKay's work showing it extends lifespan, that's not new. And the complement system's role in aging and inflammation. People like Weiss Corey were talking about that back in the early 2000s. So isn't this sort of building on a well-established foundation?
You're absolutely right to bring up the history. The novelty isn't really that CR works or that complement is involved in aging. We knew those things. The novelty is pinning down C3a as this specific, potentially druggable checkpoint, linking CR's effect to reduced inflammaging in humans, and then validating it with an inhibitor in mice. For biotech, that's important. It potentially de-risks the search for CR mimetics. Instead of vaguely mimicking CR, now there's maybe a clearer molecular target. That's a big step for drug discovery.
Right, that distinction's key, moving from a broad concept to maybe a precise lever. But it does raise the big question, doesn't it? Can C3 inhibition without CR give similar benefits in humans? Because if it can, that bypasses the huge challenge of getting people to stick to strict CR. What are the hurdles there, translationally?
Oh, absolutely. That's the million-dollar question for anyone looking at this therapeutically. I mean, the complement system is vital for immunity, right? So messing with it, that carries risks. Immunosuppression, other off-target effects. We need to know, is C3a really a central hub for CR's effects? Or just one piece of the puzzle? And the clinical meaningfulness, extending mouse lifespan by weeks versus translating that to years in humans. Big difference. Biotech needs to see pretty large effect sizes and a clear path forward to invest the kind of money needed. So yeah, mechanistically interesting, definitely. But the next steps are rigorous safety and efficacy trials.
Okay, so a very solid paper, mechanistically quite intriguing. It refines how we think CR works, points to a specific target, C3a, but still needs a lot more validation to see if it's a truly impactful target for human health span beyond CR itself. Okay, building on that idea of systemic impacts, let's shift focus. How might brain function, like cognition, tie into something seemingly far off, like our gut? Next up, we've got a paper playing into that popular gut-brain axis narrative. Title is TSP50 in Neural Stem Cells Regulates Aging-Related Cognitive Decline and Neuroinflammation by Altering the Gut Microbiota.
Right. This one's from Yao Li, Yukong Chen's Wei Gao, and their team at Northeast Normal University. PubMed, August 5th, 2025. They found something quite interesting. This protein, testes-specific protease 50, or TSP50, its expression was way down in the hippocampus of aged humans and mice. And more strikingly, if they knocked out TSP50 specifically in neural stem cells, NSCs, in mice, it seemed to accelerate aging. The mice showed worse cognitive decline, memory, learning issues, and more neuroinflammation. It directly links TSP50 in those brain stem cells to systemic aging signs.
Wow, okay. So TSP50 and NSCs seems critical. What does that mean for the bigger picture of brain aging? Do they dig into the mechanisms?
They did. The paper shows that these mice, the ones lacking TSP50 in their NSCs, they also had impaired intestinal barriers, basically, leaky gut. Plus, significant gut microbiota dysbiosis, an unhealthy mix of bacteria and lower levels of short-chain fatty acids, which are important metabolites from healthy gut bugs.
Mhm.
And here's the real kicker. They could actually rescue some of this. Restoring the gut microbiome using fecal microbiota transplants, FMT, or separately just overexpressing TSP50 back in those neural stem cells. Both approaches helped alleviate the cognitive decline and neuroinflammation. It really points a finger at the gut as a mediator here.
The gut microbiome. It's such a hot area, but sometimes feels, well, heavy on correlation, light on clear causality. What makes this TSP50 paper stand out for you? Why isn't it just another correlative study?
Yeah, I confess I sometimes get a bit wary of gut effects brain papers. There are so many. But what I think is genuinely novel here is the attempt to draw a direct mechanistic line from neural stem cells via TSP50 to the gut. It's not just vaguely saying the microbiome matters. It proposes a specific molecular and cellular pathway. It shifts things potentially from just correlation to a specific molecular bridge. That opens doors, right, for intervening in brain aging via this novel gut connection.
That's a really important distinction, TSP50 as a potential molecular bridge, but TSP50 itself isn't new, is it?
No, TSP50 has been studied before, mostly in cancer research, actually. Since the early 2000s, it's involved in things like cell proliferation, apoptosis. So its role here in NSCs and linking to the gut, that's the novel context, the rescue with FMT. Well, it's encouraging, but frankly, almost expected in this field now as a proof of concept.
Okay, so for biotech thinking about this, the big question is, will boosting TSP50 or tweaking the gut alone be enough to really move the needle on health span or cognitive function in people? And is this pathway even conserved in humans? What kind of effect sizes are we seeing in the mice? Are we talking weeks, months?
Exactly. Those effect sizes are crucial. Weeks of cognitive preservation in a mouse. How does that translate to meaningful years for a human? That's key. For biotech, yeah, targeting TSP50, maybe with a small molecule, or developing very specific gut interventions for this pathway. It's intriguing. Early days, though. And, the path to a clinically viable FMT product is way more complex than for a typical small molecule drug.
So an interesting angle on a familiar theme. It highlights a specific potential link between brain and gut health,
Yeah.
even if it builds on existing knowledge. Definitely one to watch. All right, finally, let's get into something really fundamental to aging. Genomic instability, specifically heterochromatin loss. The last paper is titled Glial Reactivity and Cognitive Decline Follow Chronic Heterochromatin Loss in Neurons.
Yeah, this one dives deep. From A.G. Newman, J. Sharif, P. Bessa, and colleagues at Charité in Berlin, published on PubMed August 9th, 2025. What they showed is that when neurons lose their ability to maintain heterochromatin, that tightly packed DNA, and when endogenous retroviruses, ERVs, get de-repressed, it leads to a chronic inflammatory state in the brain. And that drives neurodegeneration and cognitive decline in their mouse models. It's a direct link. Genome integrity breakdown to brain aging.
Okay, let's unpack that a bit. The ERV activation connection. That's really interesting. It ties epigenetic aging changes in gene expression, not the DNA sequence itself, directly to innate immunity. Essentially, these old viral bits in our genome, normally silenced, get reactivated as the chromatin loosens with age. And the immune system freaks out, seeing them as invaders. This definitely builds on work from like the Sinclair lab and others, showing heterochromatin loss as a hallmark of aging. They seem to be digging into the how.
Exactly. The level of mechanistic detail here is quite impressive. They pinpointed specific roles for HP1 proteins, HP1 seems necessary for laying down H4K20Me3, that's basically a molecular keep quiet sign on the DNA. And HP1 deficiency messed up DNA methylation, which also affects how genes are packed. When they lost both HP1 and HP1, they saw specific methylation changes right at these ERVK elements. That led to the ERVs getting switched back on, driving chronic inflammation and cognitive decline. It really specifies which epigenetic parts are failing and what happens next.
That is a strong mechanistic story. But the critical question, as always, is this a primary cause of aging or just a downstream consequence? Couldn't this ERV activation just be happening because of other aging processes causing genomic instability? How do we separate cause and effect here?
That's the perpetual challenge, isn't it? Proving causality is tough. Ideally, you'd want an intervention that reverses the heterochromatin loss and see if that reverses the aging phenotype. This paper shows losing it causes problems, but not definitively that restoring it fixes everything in the same way. So the big question for biotech becomes, can we actually maintain heterochromatin structure? Maybe through things like NAD plus boosting, which has shown some preclinical promise in epigenetic reversal. Can that actually prevent cognitive decline via this mechanism? That's the translational hope.
But even if it's not the absolute primary cause, couldn't this ERV activation still be really important, like a canary in the coal mine, a clear signal of genomic chaos that could be a valuable biomarker, or maybe even an intervention point itself, even if it's downstream? For a biotech, wouldn't a good biomarker for early brain aging or neuroinflammation be incredibly useful for trials, regardless of ultimate cause?
Oh, absolutely. You raise a great point. Even as a consequence, it's a damaging one. It perpetuates inflammation and degeneration. So, yeah, interrupting that cycle, resilencing ERVs, stabilizing heterochromin, whether with small molecules, gene therapies, maybe even NAD plus precursors, it's still a compelling therapeutic strategy. The challenge, of course, is specificity. How do you target just the ERVs without hitting essential genes? How do you get durable heterochromatin stability without off-target effects? It's complex epigenetics. Developing drugs here is still frontier science.
Which brings us back again to human validation. Is this a real mechanism we can target effectively in humans to improve brain health? Or is it another biomarker that looks like a driver but isn't the main one? That distinction is critical. Yeah. So looking back at the week, as you can see, these papers, while definitely thought-provoking, giving us good mechanistic details, they largely feel like incremental advances. Solid refinements, deepening our understanding, but maybe not the paradigm shifts rewriting textbooks tomorrow.
That's a fair summary, yeah. Even the complement pathway paper, which is probably the most promising in terms of a specific target, it still needs significant validation. We need to see if targeting C3A can really offer benefits beyond what we already get from CR and if it's safe long term. The findings are exciting, yes, but that translational leap, it's still pretty large. Needs serious investment to de-risk.
It really just underscores how science usually progresses, doesn't it?
Mm-hmm.
Steady accumulation of knowledge, gradual refinement of targets. Not always those sudden revolutions. It's often about finding the precise molecular handles within those broader, known hallmarks of aging.
Which leads to a question for you, our listeners, particularly those in biotech R&D. Given this typical incremental pace, rigorous but incremental, what do you see as the biggest hurdles right now? Hurdles in translating these kinds of promising mechanistic findings, these specific targets and pathways we've talked about, into real, clinically meaningful longevity interventions for humans. How can we collectively accelerate that translation, moving from intriguing mouse data to scalable human health benefits? Something to think about.
Thank you for joining us for this critical analysis of the latest research. Keep thinking, keep questioning, and keep exploring. We look forward to digging into the next batch of longevity papers with you soon.